Okay, so we're going to talk about transitioning from drug-centric to patient-centric trials, and I think it's a really good follow-up on Dr. Churchick's work. But I'll start with a little bit of an overview, a talk about that, and then we have our panel. But I would start with introducing the panel. And Dr. Young Che, do you want to introduce yourself to start?
Yes. So my name is Young Che, from Chicago. I work at Northwestern. I'm co-director of the Developmental Therapeutics Institute and director of the lung cancer program. Also co-lead the SWOG Early Therapeutic and Rare Cancer Committee with Dr. Kurtzrak. I'm the vice chair at SWOG, and also been involved in running the rare cancer trials with her, especially through the DART SWOG 1609 program.
Thank you. And Jim, you hardly need an introduction, but if you can introduce yourself regardless.
You've already heard from me, but again, I'm Jim Palma. I'm the CEO of TargetCancer Foundation, and I'm up here today to talk about the TRACK clinical trial, which is a decentralized precision medicine clinical trial in rare cancers that's being run from TargetCancer Foundation.
Jen?
Jens Ruder. I am a medical oncologist by background. I'm the chief medical officer at the Jackson Lab, which is actually a non-profit basic science research organization, but we started a clinically focused program in the state of Maine, where Jax is at home, so just a little bit north of here, to really bring precision oncology to community setting in a very rural state. And also, so we've done this now for about 10 years, and I think that's my role in this panel.
So I'm really grateful to the panelists and the work that they've done to try to further patient-centric trials, and especially in rare cancers. And so that's what I'm going to talk about, drug-centric to patient-centric cancer therapy, and tumor-agnostic to N of 1. Basically a new paradigm, treating each patient as an individual. So the way we do clinical trials is the drugs are at the center of the universe, and we try to fit patients onto drugs. And we need to change that and put the patient at the center of the universe and fit drugs to patients, not the other way around. And so this is my disclosures. And just a little bit of background about what I've done before and how that's informed my thinking. I was the founder and director of the Center for Personalized Cancer Therapy at University of California, San Diego. And before that, I was the founder and chair of the Department of Investigational Cancer Therapeutics, which is the phase 1 department at MD Anderson Cancer Center. And which grew to be one of the largest, if not the largest, phase 1 in an academic center. And the reason I want to emphasize this is because this has given me a lot of opportunity to look at molecular typing. And in addition, sometimes when I talk about what we're doing, since we're also trying to break paradigms, people say, well, you don't know anything about phase 1. And well, yes, I do. And I want to emphasize that maybe that the way we've always done phase 1 is not the right way, and we need to rethink it. One of the poster child for a patient-centered trial, I think, is the TargetCancer TRACK trial, which is a clinical trial completely remote that provides genomic information and molecular tumor board-based treatment recommendations for patients with rare cancers. It's totally patient-centered. It's totally N of 1. Each patient is considered as an individual, and we consider it a home run trial, playing on the words of home run, but that patients can stay at home. And Jim mentioned this before, but we have patients from 44 states. It's an umbrella trial across the nation. I think over 400 patients have consented to the trial at this point in time. So take-home point number one, cancer is a disease of the genome. And this is, we need to completely rethink, I believe, how we think about cancer, which historically we've thought about it by organ of origin, and we still do, but it's really a disease of the genome. And this is a very difficult transition, not because of the science, but because of the constructs that we've made where we work by organ of origin. But I think only by understanding that it's a disease of the genome and treating accordingly can we really move ahead where we have responses like Dr. Churchick showed in almost 100% of patients. So you'll see Dr. Sabai and I have written multiple papers together, but really genomics is the diagnosis. So what we believe is that the diagnosis is not necessarily colorectal cancer or lung cancer or breast cancer, but the genomics defines the diagnosis of the patient. So what do we do when we see cancer patients? The first thing we do in order to treat them, just going to put this down, is we try to diagnose them. And the way we diagnose cancer patients is with the light microscope, which was invented in 1590. And this tells us where the tumor came from. But it does not tell us what is wrong with that tumor. So essentially, by just knowing where the tumor came from, we're missing the target in cancer therapy. And we are now beginning to look at a reclassification of cancer based on the molecular microscope, what I'm going to call the molecular microscope, which is genomic sequencing. And there's been breathtaking progress, unparalleled in human history, to be able to use this technology. In the 1990s, it was thought that the human genome could never be sequenced. By 2001, never was over, and we had sequenced the human genome. It cost $3 billion. By 2007, it cost $100 million, still a little bit high for insurance to cover it. Now we're talking about the $100 genome, and really every patient can have their tumor sequenced. So this allows us to find the target. And this has allowed us to redesign cancer clinical trials. And the first stage of that redesign was to understand that lung cancer is not one disease, but multiple diseases defined by their cohorts, by their genomics. So it could be ALK fusions, it could be EGFR mutations, and so forth. And then we moved on to the next stage, which was a real jump, to understand that the same genomic alteration in multiple cancers could have the same treatment. This is the tumor agnostic treatment. So if you had an NTRK fusion, or if you had a BRAF V600E, no matter where that tumor came from, you could have the same treatment for that patient. This moved us from large randomized trials of unselected patients where only a few patients would respond to very small trials. Often FDA approvals are on anywhere from 30 to 100 patients, but the work went on before the patient got on trial to try and find those patients. NTRK fusions, 1 in 300 patients, but 75% of the patients will respond to an NTRK inhibitor. And this allowed multiple tumor agnostic approvals. So the first tumor agnostic approval, which relates back to what Dr. Churchick does, was for pembrolizumab and anti-PD-1 for solid tumors based on MSI high. And this really shattered the paradigm. Really, congratulations to the FDA for being so forward-thinking. It was a tissue-agnostic approval, and it was approval of an immunotherapy based on a genomic marker, which was MSI high. At that time, it was for patients with very advanced disease. And even in patients with very advanced disease, a substantial portion of them responded, and a subgroup went into complete remission and might have been are cured. Of course, not the huge subgroup that Dr. Churchick sees now with moving it earlier, but still, it was amazing to see patients with advanced disease cured of the disease. So we now have 10 tumor agnostic approvals from the FDA, and although each of these groups are rare, when you put them all together, that's not so rare. Not that being rare should be an excuse for not finding the alteration. It should never be an excuse for not finding the alteration. But the claims that, oh, these are each rare. Yes, they're each rare. But then when you put it all together, you have to do genomics on these patients because the percentage of patients in every cancer becomes more substantial if you look for any one of those 10 alterations. And this is especially true and important for rare cancers where there's often not a good treatment. Okay, but that is just the beginning. And take-home point number 2 that I want to emphasize is the next stage, going from these rare alterations to ultra-rare to N of 1, to considering each patient as an individual. So how do we determine therapies? The traditional way of determining therapies is finding commonalities between patients to decide therapies. At first, it was tumor of origin. For example, are you breast or endometrial or lung cancer? And then it was molecular marker alterations within tumors. Do you have an EGFR mutation? Do you have a lung cancer, or so forth? And then we evolved to tumor agnostic markers. You do have an NTRK fusion, but I don't think that's the end of the evolution. I think the next stage in the evolution is to understand what if every metastatic cancer, and really every cancer is different, like malignant snowflakes, each is complex and each is distinct, and we need to treat patients accordingly. So here is an example of next generation sequencing done on a series of breast cancer patients, and we've looked at thousands of patients. This is through across thousands of patients, but the slide doesn't cover that many patients. So here we have a patient with an ERB2 or HER2 amplification. We have another patient with ERB2 or HER2 amplification, and these 2 patients would get the same therapy. But if you look at this, that can't be quite right, because each patient has more than one alteration. And what we really need to be doing is treating all their alterations, not to be treating them with one drug just because they have something in common. And then the second part is that the alterations differ from patient to patient. So they shouldn't be getting one therapy. They should be getting a patient-centered set of drugs that is formed for their cancer, is customized for their cancer. So we shouldn't be playing a whack-a-mole game. The next stage, I think, is N of 1 patient-centered clinical design, where we go, number 1, beyond genomics to multi-omics interrogation and personalized combination therapies. And this moves us from a drug-centered universe, which I mentioned before, where the drugs are the center of the world. We find patients for their just drugs, instead to a patient-centered universe where the patient is the center of the universe, and we find drugs for the patient. So just the opposite. And this requires that we create new clinical trial designs that emerge from the molecular reclassification of cancer. So we've been working for several years. This study started a study called iPredict, Investigation of Profile-Related Evidence Determining Individualized Cancer Therapy. To do such a trial, the study was IRB approved about a decade ago, looking at histology-independent targeted approach, looking at multiple molecular alterations, and patients are individually matched with a customized therapy. And the idea was to treat patients with aggressive malignancies, do molecular tumor board, have a matched therapy, and then see how well patients did depending on the degree of matching, and every patient got a customized therapy. So overall, in these pretty heavily pretreated metastatic patients, what we saw is the better the matching of the customized combination to the drugs, the higher the response rate. So these are patients that were better matched, and this compares to patients that are poorly matched. And when we looked at progression-free survival and overall survival, the results were reflected that as well. So in highly matched patients, the 2-year survival was over 60%. In patients that were low matched, the 2-year survival was under 20%. Now, what are some of the issues with doing this? If there are 300 drugs in oncology, there's 45,000 2-drug combinations and over 4.5 million 3-drug combinations. So that's a lot of phase 1 trials to do. You can't do them. And if there's 700 cancer gene and the median number of alterations per patient is 5, there's over 1 trillion possible molecular patterns. So what we really need is some sort of artificial intelligence, machine learning to understand the best pathway for each patient. And complexity is why single agent therapies are generally matched, because it's hard to do it any other way. But I think that's now we have better technology and we need to move on. And that's what we tried to do with the iPredict trial. Now, one question that comes up is, if every patient gets a different drug regimen, what is the endpoint? Because you can't say that the endpoint is drug A and B in tumor lung cancer, because everybody's getting a different regimen. So it's really looking at cancer in a different way, and you analyze and approve the robustness of the algorithm that matches patients to drugs, not the individual drug combinations. And this treats every patient as a unique patient, which they are, and customizes therapy to them. So the trial was published in Nature Medicine, featured in National Geographic and the NIH Director's Blog, and I was honored to present it at the Nobel Symposium for Precision Medicine as well. So here is what I will finish with. The problem is current phase 1 trial paradigm. I mentioned that I was a phase 1 doctor. And what I'm going to talk about is the paradigm for novel combinations of approved drugs. And the paradigm has been that you can't do novel combinations without a phase 1 study. But I just mentioned that there's 4.5 million possible novel combinations, and we're not going to be able to do phase 1 trials for all of that. So how did we overcome that in iPredict? And this is a quote from Dr. Sabaya, but it is not the mountain this is from actually Sir Edmund Hillary, but Dr. Sabaya introduced the quote to me. It is not the mountain we conquer, but ourselves. So Edmund Hillary was the first person to climb Mount Everest, and I think this quote really applies to us as oncology researchers. We need to get over our structures that we've imposed on ourselves. So the current phase 1 trial paradigm for dose finding is we do 3+3 design, 3 patients per dose level, or accelerated design with 1 patient per dose level. We find the maximum tolerated dose on 6 patients, and each trial takes at least 1 to 2 years to complete. So you can see that there's problems for novel combinations. Like the first problem is we're actually finding one-size-fits-all dosing based on very small numbers of patients, which is kind of crazy. And then it takes phase 1 trials at least 1 to 2 years to complete. We can't do 4.5 million phase 1 trials. That's impossible. So I mentioned this number before. And also, if you really think about it, what are we doing? These 2 patients both have cancer. I pulled them off the internet, but they both have cancer. Does it make any sense to give them the same dose? I don't think you need to be a physician to know that it doesn't make any sense. And what dose does she get? She's actually my favorite person. So the take-home point number 3 is that personalized dosing is critical. And Dr. Nikunjam and I did some work looking at personalized dosing, finding some rules for personalized dosing. And that's how we did the iPredict trial. We started at lower doses and we personalized the dose for every patient. And what we discovered was that's not just good for new combinations, it's actually good for everybody if we can personalize the dose, because we shouldn't be giving 20-year-olds and 80-year-olds the same dose anyways. So looking into the future, which is what I'm going to wrap up with, is some of the trials like iPredict allow us to do novel combinations in real time for patients. So it's discovery of real time, treatment in real time. We don't have to wait 2 years to create a new protocol. We customize the therapy for the patient that we see in the clinic that week. And we can get the therapy ready for them in just a couple of weeks. And this is an example, was an international collaboration, but I'll finish with a patient that I think is a really good an example. This was a rare cancer, fibrolamellar cancer. They have DNAJB1 PRKACA fusions. These fusions are undruggable. The patient had was biomarker negative for immunotherapy. The patient had a low tumor mutational burden, PD-L1 negative. The patient had already failed an anti-PD-L1, which is atezolizumab, and had this undruggable fusion. But we worked with this company called Pangea, which does RNA synthetic lethality. And their prediction, which is an AI algorithm, was that the patient would nonetheless respond to Nivo and Ipi, and the patient did respond. And at the time that we published her, she had been told that she was going to hospice, but now she was 2 years in complete remission, and it's now 5 years, and she's still in complete remission. So she's probably cured of the disease, which I think speaks to the potential of treating patients as individuals and using AI-based algorithms to decide their therapy successfully. So I just want to end by thanking the patients, and especially in our precision medicine team. And then I'd like to go on to the panel, and if you can speak a little bit about the patient-centric trials that you've been involved with, and then if there's questions from the audience as well. Dr. Yang-Che.
Yeah. So, yes, thank you for your wonderful talk. And I am, on the same page with everything you mentioned. And I'm so thankful that because you published your effort, iPredict, and, in a more concerted, systematic way than what we've been doing in practice. Because as a phase 1 doctor, I've been doing a lot of the off-labels. There are trials, but a lot of times there aren't good trials, and they come and go, they close, and they temporarily close. So we come up with the best rational combination, whether it's through molecular tumor board or just us looking through literatures, or nowadays getting help from the AI platform out there. So we are doing that, and luckily in the US we are able to, not 100%, but get these novel combination, and sometimes those lead to new clinical trials. And then we do report, like you presented your last case report, we do find unusual responders, and that leads to another breakthrough. So I do think patient-centric approach and looking at the molecular profile to really tailor the best combination for each patient at each individual level is a very important approach. So yes.
Okay, thank you. And I'm going to come back to you because I also want you to talk a little bit about the DART trial. But before we do that, let's go on to Jim. And if you want to speak a little bit about whatever you want to speak about, maybe the TRACK trial.
Thank you. So we took an approach at TargetCancer Foundation to creating a patient-centric trial that was really informed by our role as a patient advocacy organization, and it was developed in response to what we were hearing from the patient community, which was a lack of access to clinical trials, certainly a lack of access to biomarker testing, and for patients who were getting tested, not correct interpretation of those reports when they came to them. And, as you heard this morning, we were informed by an experience with cholangiocarcinoma, which is a perfect example of this, where patients were routinely put on a standard of care chemo regimen while there were FDA approvals happening for targeted drugs for IDH1 and FGFR fusions. And day after day, we were seeing patients who either never were tested for these, or in some cases even were told that they had these fusions or mutations, but were never given a targeted therapy. So TRACK was really built in response to that problem, and it has dual or sort of parallel patient-centered and research-centered goals. So on the patient side, it is a fully patient-driven trial. Patients come to us directly and contact our team to enroll. And when they do that, they're given the opportunity to consent remotely through an e-consenting process. All of this happens from home and really happens independently by the patient. Once they consent, we do all of the work on the back end to collect medical records, to facilitate the transfer of tissue to Foundation Medicine for testing. And also we send a mobile phlebotomist to their home to do a remote blood draw so that, again, really the patient can stay at home for the entirety of this. And a key element of this was that the foundation testing comes to the patient at no cost, because cost was also a barrier that we were hearing about. When the reports come back, we package the reports along with a medical history that we've extracted, and we present it at our weekly molecular tumor board meeting, where we have people like Dr. Kurzrock and many other people in the room here who are able to provide interpretation and provide recommendations for treatment, often for the types of combination therapies that Dr. Kurzrock was talking about. And really this was in an attempt to alleviate that other issue that for so many patients, they may be seeing a physician who's never treated their rare cancer before, and certainly couldn't be expected to know the full treatment and clinical trial landscape for whatever is being determined by their genomic testing report. So we provide those recommendations back to them. And really at that point, it's up to the patient and doctor to decide if they want to accept those recommendations or not. And I think we'll speak a little more about the challenges there. From the research side, we're of course creating a lot of genomic data in cancers, over 65 rare cancers that don't get a lot of attention, and we'll be publishing on that hopefully very soon. Developing a decentralized trial, in my opinion, is the most patient-centric way to do this. The benefits that we've seen have been amazing, and we experience that every day with the ease by which patients can come into the trial. It also comes with tremendous challenges from a logistical and operational perspective, which I think is what limits broader adoption. And we can talk more about that.
Yeah, and if I can make a comment, Jim has been amazing in being able to operationalize it. He makes it look easy, but it's actually not easy to operationalize a trial that is across the entire United States. So very grateful to be able to work with you on it. So Dr. Ruder, do you want to speak a little bit?
Sure. So we have, through the Main Cancer Genomics Initiative, which started back in 2016, our primary goal was to really enable the community oncologists and rural cancer patients and rural doctors to take part in this revolution, as Dr. Kurzrock outlined. The approach, so we actually, even though I think it's a very patient-centric approach, we actually focused on enabling their physicians. That was sort of the approach that we took. And we did this through two different mechanisms that have sort of shifted in relevance over time. We started out by providing the testing free of charge, similar to the TRACK study. We have a clinical lab at the Jackson Lab that actually performed the testing. That was back in 2015, 2016, when actually getting testing was still a very, very major barrier. I think actually that issue has, and we can certainly debate this, but has to a huge part been resolved, as there are many different testing providers out there now that do this. And I think the patient out-of-pocket costs are are relatively manageable at this point, although again, there are going to be outliers. The second most important aspect of our program, though, was to help physicians better understand the testing so that they could actually explain it to their patients and then also personalize the treatment for their patients. So we also implemented a molecular tumor board. We call it a genomic tumor board, but it's essentially the same concept, which also strives to really look at the individual profiles and identify treatment options. Now, I think Dr. Kresrock's approach that you presented here is certainly very incredibly innovative, and that's an area that we're now also starting to look at, because I do think just playing whack-a-mole with one target at a time may be very difficult. But ultimately, I think we have to start somewhere, and our initial goal was really to identify what are the on-label options. Very straightforward, you would think, but sometimes it's actually somewhat hidden for physicians to understand. And secondly, what is the rationale for using a drug off-label? And thirdly, and we should not forget this as well, are there clinical trials, and where are they, and are they available to patients? So I think we've been quite successful in this program in the last 10 years. We have also published our experience with this. Interestingly enough, we found that in the first phase of the program between 2016 and 2020, which enrolled about 1,500 patients, that around 16% went on a therapy that was matched to their profile, which really tells you where I think partially the problem is. I think that number has to, go up, because there's certainly more targets that are available. But interestingly enough, even that 16%, that small population, had a better 1-year overall survival than the population that did not receive a matched therapy. Again, providing some additional evidence that even if you do this in the community setting, you do improve patient outcomes. One last thing on that is that we also the patients were all enrolled in a study protocol, and so we're actually able to ascertain what are patients' perspectives, expectations, hopes, and this type of an approach, and we've published a number of publications on that as well. So I think there's more to come. There's more work we can do, but certainly it's a good start.
Thank you. If there's questions, we're happy to take them. But in the meantime, I wanted Young Che to mention the DART study, because it's an immunotherapy study aimed at rare cancers. And took a little bit of a different track, but if you could give an overview of that study.
Sure. So we met in 2015, talked about rare tumors I guess the immunotherapy don't discriminate rare tumors. And at that time, we had nivolumab, ipilimumab data for melanoma. And then there are rare tumors like Merkel cell, anal cancers, like showing Hodgkin's lymphoma, showing good responses. So we thought about a trial where we can lump all the rare tumors together as a one basket trial and deliver combinational immunotherapy. So DART stands for anti-dual PD-1 CTLA-4 blockade for rare tumors. And 2017, we activated the trial, was open through the all NCTN, was led by SWOG, myself, Dr. Kresrock, Dr. Patel, were study chairs, and we worked closely with the NCI, and it was open through the whole NCTN network, so it was open more than 1,000 sites. We ended up accruing close to 800 patients over more than 6 years. We are now close to new patient enrollment, but there are still patients on active therapy still now. And what we learned is that rare tumors, we've seen deep and durable responses in most of the cohorts. And it's just up to us. We are doing the translational research to really find out who they are. But in some cohorts, we have cohorts like high-grade neuroendocrine tumors where they could be of any tissue origin. And we've seen very good response rates and very good durable responses. And a lot of them have also led to update in NCCN guidelines. So the Nivo plus CP regimen has been in the NCCN, allowing patients to really get it regardless of the FDA approval. A lot of American insurance companies look at NCCN guidelines, so we do think it's practice changing. We're still learning. So I think going forward, what we're thinking is we need platform trials like this for rare tumors. And unlike molecular targeted therapy, I was one of the PI for the NCI match, which was a preceding trial, ended up being enrolling about up to 30% whole rare tumors. It was a pan-tumor trial, molecularly targeted, had close like the 30-40 arms in total. The DART trial had up to a total of 53 arms, was all different rare tumor histology. We even had non-otherwise specified. And then we were enrolling cancer types that we never heard of and ended up making a separate cohorts for them. We had PDL1 and copy number amplified cohort. So we were very innovative. We were adding cohorts as we were gathering more data, like the hypercalcemic small cell ovarian cancer was one cohort we added when we knew that histology was specifically sensitive to immunotherapy. So it was a pretty adaptive trial. I think we had like 10 revisions along the way, along that 6 years. So we were able to adapt to new information and really accrue well. And we've shown that, yes, through a lot of these accruals happen through the community sites. So more than half of our patients were coming from the community sites, and the trial speaks for itself. That yes, we can accrue to rare trials when we do it as a basket trial, and Dr. Kudrick and I are really working hard to make it like a platform trial where we need a second drug in place. So we are working hard to make DART2 the next available trial, but stay tuned.
There's a question over there. Do you want to go ahead? And if you could introduce yourselves when you ask the question. Some of you I know, but I don't know if everybody knows you.
Jesse Bohm. The concept of a machine learning classifier that can take the molecular information from a single patient's tumor and make predictions is clearly a goal for all of us for the next 20 or 30 years. But it's predicated on having data that can guide such predictions. And it's not clear to me if such a data set exists at the right scale internationally, where is it? And if it doesn't exist, what are its attributes so we can work together to make sure it's generated?
So I think the data probably does exist. The question is the access to the data. The amount of data that is now available is almost unimaginable through electronic health records and so forth, and also through publicly available data like TCGA. It's making it accessible, and even more important, this is kind of breaking the model. And so for the FDA or NCCN to approve a prediction algorithm is completely different than approving a drug combination for an individual disease. So I don't know who's going to shatter that paradigm, but I think that's actually the more important part of it. More important than the data availability is how do you get this to an approval? And by the way, it also means that the algorithms will predict what you don't respond to. So it's not just about saying this is the best drugs for you, it's about which drugs you shouldn't get as well. But I don't think it's a data problem at this point. I think it's a model problem.
But in citing TCGA, I think you're suggesting that if we had an infinite amount of genomic data, even the absence of clinical data over drug response data, that would be sufficient. We know if that is the case, or other types of data that have drug response.
I think what we need is data on genomics and actually multi-omics and corresponding data about responses. And many of the big companies have that data in their databases. We know that companies like Foundation Medicine, Tempus, Caris have tremendous amount of data in their databases. I don't know the extent to which they're working on an algorithm. I have to believe that they are. But again, my sense is this is no longer a data availability problem. It's that we have to make a big switch in our thinking, and that's hard to do. Did you?
Yeah, I'm Jeff Waldron, and I'm bringing one of my areas of focus in, which is kind of an extension of what you're doing, which is working with pharma companies on their expanded access programs, whereby patients with advanced disease can get investigational meds before FDA approval. And I've been working hard with them because, that's going to be the target patient population once the drug is approved, whereas the trial patients are often earlier stage disease because they don't want to jinx the approval process. But I see your sort of patient-focused approach moving in that direction of expanded access, where patients who are no longer responding to approved drugs can more quickly get access to investigational therapies. I just want to get your directional sense on that.
So I think that's a very important direction. And back in about a decade ago, we wrote a paper in Nature Reviews Clinical Oncology about right to try. I actually wrote it with my son, who is an attorney, and my husband, who's a physician, but we were very strong advocates of right to try, and the right to try legislation was passed. But I think what you mention is pretty important. I think that there's hesitation on the part of drug companies because, and I can actually kind of understand this, they think that their most important goal is to get that drug approved. And from a patient point of view, I have to agree with them. And then if the drug gets into everybody's hands and then there's side effects, they're afraid that that will upend their FDA approval. So I can actually kind of understand what they're saying. If we want to get these drugs to the patient as quickly as possible, it's an experimental drug, we can't upend the approval. So I don't know what the answer is. Maybe the answer is that the FDA will guarantee that they won't consider these compassionate ad hoc side effects. But I will say that the compassion INDs, though most people don't know them, they're actually really, really easy to get. We just got one on a 34-year-old man, and it took us about 2 weeks to figure out who knew how to do it at our institution. But when we figured it out, we had the drug in 24 hours. So it's been amazing legislation. I don't know if anybody, do you have any comments about the compassionate?
Maybe not directly on that, but the issues of access for patients with rare cancers is critical, and we experience this in our trial very often, where there's a number of reasons why treating physicians might not accept a recommendation that our MTB is making. But feedback that we hear very often is, I'm not going to offer my patient a drug that they can't get, whether that's because they're anticipating insurance denial or the difficulty of receiving a drug through compassionate use. So I think developing pathways for patients in general, but certainly within this community, to help patients access these drugs and help physicians understand what you were just talking about, that this isn't always so difficult to do, to access them, could be really helpful, because it's a shame to see that that attempt might not even be made.
So I think accessing the drugs is a really big barrier. And at our institutions at UCSD, now at MCW, we set up a system to access the drugs, and it consists of having a medication acquisition specialist. And at MCW, that person is a PharmD who's actually working now with the Dr. Reuter's group as well. And he works in clinic with us, and he can, I call these people magicians. They can access almost anything because there are methods to do this, legal methods to access drugs. But the thing is that our methodology is not scalable. Ultimately, we can produce data, but ultimately it's only scalable through FDA or NCCN approval, so that people in the community or at other centers can do it without spending hours trying to figure out the pathway. Dr. Subbiah, you had a comment.
Yeah, no, absolutely. I think that's a great question. I completely agree. A major believer of compassionate INDs. main pushback is from institutions because they think it's a lot of paperwork. And I filed a zillion compassionate access protocols, but it increased administrative burden, paperwork. Fortunate enough to get some philanthropy funding for staff to take care of it, but it's always a challenge to get it through the institution. It's easy to sometimes even get the drug. FDA approval is, I got it in 1 hour. An email came back from FDA, okay to use in 1 hour, right? It's mainly the institution and the oncology colleagues who say, hey, when you're out, I'm not covering for this. I'm not taking care of this. So that's been the challenge. Resistance from not the cancer, resistance from the infrastructure, the institutional resistance. That's been the challenge for all these compassionate access protocols. Another thing what we've done is many times if there is a molecularly targeted drug and if the phase 1 is ongoing, it will be at a low dose. It will take 2 years for us to get to the a ready dose. So we've asked the company to do an NF1 customized study where we do intra-patient dose escalation. And we proven, and we showed responses, and we even published those in NF1 cases. And in fact, the company could fast track the phase 1 trial, which was supposed to be going on for 2 years, in 6 months because we filed 4 compassionate access. We did intra-patient dose escalation study. We found the dose in those 4 patients and gave it to the company. And then they amended the protocol to add that data to get that data to the phase 1, ongoing phase 1 study, and they got the RP2D.
So I think you're 100% right. And really, like 15 years ago, getting up compassionate access took hours of work. When I was at MD Anderson, I had to tell our regulatory person that they had no work to do for a week, but to get this patient compassionate access. But since the right to try legislation, as Dr. Subbiah said, it's really easy, but it's so easy that it's hard to find somebody who knows the pathway. And then everybody thinks this is way too easy. So our IRB tried to get involved and the FDA said, quiet. This has gone through. The FDA is actually supportive, but everybody's thinking that there needs to be more paperwork here. So people don't realize even I didn't realize how easy it was. Did you have a question? Somebody else?
Yeah, back here.
Yeah.
I'm Jeff Kramer with the Chondrosarcoma Foundation. And just to talk about expanded access and compassionate care, in our experience, it's like an act of Congress to get approval. First off, the burden on oncologists to fill out the paperwork that's necessary, and the fact that their caseloads are so high, many of them are very reluctant to even go through that. And we particularly have problems with terminally ill patients, those who are terminal. It's like you're too sick to even bother with, So we've encountered lots of barriers. And go ahead.
What I'd like to say is that I think the barriers are less than people think they are, but you have to have a person in the practice that knows what to do. And very few practices or even academic settings have an assigned person to do that. And the physician with 20 patients in the clinic cannot do it. Somebody has to know the pathway. And I don't know, Dr. Reuter, if you want to speak to this from a community setting. We've been doing the molecular genomic tumor board for a while, and I don't know if there's feedback about how to get the drugs, because I see that as a barrier.
Yeah, no, I think that is absolutely correct. We've actually seen over the years sometimes surprising examples, I would say, where patients were started on therapies that are not directly FDA approved. Compassionate use is, I think, yet another barrier, right? We have to always be very clear. There's on-label, off-label, and then compassionate use. They're all very different categories. I would say that in community practice, you sometimes even have a hard time with the standard therapies and with on-label therapies to some extent, especially as you get into patient populations that are either have Medicaid or sometimes even Medicare. So I do think that it's still an issue. And we found, as Dr. Krezok mentioned, we started working with this medication acquisition specialist, in just one conversation sort of with the practice and with the individuals that usually manage this, I think already helps, kind of facilitate that process, because it doesn't really take that much. I would completely agree with that. It doesn't take that much. You just have to think of it. And as a physician, I think this is where it all starts, right? As a physician, you have to be comfortable with that decision. You have to trust that this is actually an appropriate way to move forward. And the more we do this more broadly in the community, I think the more likely we will get there.
So again, ultimately, to scale it, it has to be approved. But what we have is Justin, who's also working with the Maine Cancer Genomics Initiative now. He's a PharmD, and he sits with us in our clinic. And I will say, this is the drugs we want. And he'll say, okay, you're going to be able to get this drug, but I can't get you this drug because they don't have a good patient access program. Can you substitute it? And thankfully, we have a lot of redundancy of drugs in cancer. So, and he'll even suggest which drugs to substitute it because he's a PharmD. So he'll say, well, you want an EGFR antibody. I can't get you this one, but I can get you that one. Is that okay? And so the process is very fast. We're talking about 5 minutes. And then we decide on we know what we want to target, but he tells us what drugs we can get. And that way we're not going through a lot of weeks to get a denial because he already knows the process in advance. But ultimately, I don't think that's scalable. I think the approvals is what will make it scalable.
I'm fascinated. And I perked up when you talked about the patient-centered algorithms. And I'm wondering, and this might be a naive question, but what's the criteria for those algorithms? And how do you get AI to work for you in that regard?
So the criteria is, first of all, response rate and survival. And that can be looked at to see, do patients that are better matched do better? We already know they do. We published the initial results in Nature Medicine, follow-up trial in genome medicine, and we have one that is embargoed now but is accepted, a bigger trial. So ultimately, it's outcome. It's if you're better matched versus less well-matched, how do you do? And we have one paper, an additional paper that sort of looks at data in the literature, and the p-value is actually 10 to the minus 16, which is pretty incredible p-value, that the degree of matching correlates with outcome for patients. So ultimately, it's data-driven. That's the bottom line. I don't know if there's any final comments from anybody.
Well, I'll just say about the question about the AI, just one other thought that occurred to me. I do think, to answer your question, I do think that we can use AI also to help us identify potential therapeutic pathways based on specific alterations. There's an incredible amount of biomedical literature, and only sort of like the top 5 or maybe 10% of that is usually used in clinical care. So I think that's actually maybe what you're getting at as well is, can we use AI to surface that and to evaluate it for us? I think that can happen. It's just a question.
I want to add, because there seems to be a lot of interest in AI. One particular area that, we're doing within the DART trial and many other trial is through the H&E slides. So just looking at the H&E slides where everyone needs to make a diagnosis, it becomes an issue when you need extra tissue for NGS and other markers. And if you want to go granular and do multiplex IF and different markers of interest, there's not much tissue left, or there's a lot of necrosis, and it's so you actually lose a lot of people by requiring a lot of biomarkers. But since everyone has H&E slides, we're now able to train which cell, because we now have the granular data about the transcriptomics, epigenetic, proteomics, all these granular level of special information, which cells are what, even at what's regulatory T cells. So you train it's the level of the quality of data is important. So you train slides, H&E slides that has a matching special genetic or epigenetic information, and then be able to really find out are these immune cells close to tumor, and come up with a AI-based model that just with the H&E slide will be able to predict who's going to respond better, and how durable would that be, or what combination would be even better. So those agents are coming with the help of AI. We would never have been able to figure out that this would be possible. So I would just want to add that.
I think that's very important, and that's using slides that are already available. And my own feeling is that there's not going to be one algorithm. There's going to be multiple different algorithms, and they may have a different fundamental idea behind them, and they'll be able to compete with each other, just like NGS vendors compete with each other, or just like clinical trials with different drugs compete with each other. I think we should wrap up. Jim, do you have some final words for us?
No, just, I think the power of this type of conversation in this meeting is the fact that we're looking at this broadly across rare cancers. And I think any one of us individually could think about this in a particular disease area, but clearly there's a pathway here for building platforms to support this across rare cancers. That's what we want to do at TargetCancer Foundation. So, I would invite anybody to hopefully to continue this conversation, but also, speak to me or any member of our team about ways that we could work together on some of these ideas.
Jens, you're holding your side. I thought you may have some comment.
No, I mean, I think this is a great discussion, and certainly, I've not specifically focused on rare cancers in the work that I've done, but I would argue that, based on the profiling that we can do nowadays, that every cancer has become rare to some extent. So I think we all are talking the same language and thinking along the same lines. Thank you.
Okay, thank you to our panel, and thank you for all the questions.